Hello, folks, and welcome back to part 2B of our notes on gene expression. In this section, we're going to take a look at AP Bio topic 6.3, transcription. So let's just start with an overview. So what are the molecular components of transcription? RNA synthesis, which is what transcription is catalyzed by RNA polymerase, polymerase. So makes a polymer, ACE, it's an enzyme, which pries the DNA strands apart and joins together the RNA nucleotide. So it separates the DNA, unzips it, and matches up the nucleotides. Remember that RNA is complementary to the DNA template strand. Remember. And it's following the same base pairing rules as DNA, except we substitute uracil for thiamine. So if you got G on the DNA, you'll match up a C RNA nucleotide. If you got A on the DNA, you'll match up a U nucleotide, RNA nucleotide. If you have a T on the DNA, you'll still match up an A RNA nucleotide. So here we can see kind of the initiation. There's three parts to this, right? Initiation. There's the promoter region. It tells it where to start, right there. Okay, the transcription unit that we're going to read on the DNA, and we're going to read from 3 to 5 prime on the template strand down here. So we start on 3 prime, we're going to read this direction, and we're starting to transcribe the RNA. So then we go into elongation, the second step. And as we unwind the DNA, as the promoter, or as the RNA polymerase goes along, it's going to produce the RNA from 5 prime to 3 prime, going downstream. And then finally, we have termination, step 3, where it pops off, the polymerase that is pops off, and we have our completed RNA transcript. Now, if it's a prokaryote, it's done. If we're talking eukaryotes, then it's going to have to go through some RNA processing first. And here we've got this short video that'll kind of go over that, and I'll meet you on the other side of this.
The first step in protein synthesis is transcription of RNA from DNA. The portion of the DNA that is transcribed into an RNA molecule is called a transcription unit. An enzyme called RNA polymerase carries out transcription. With the help of protein transcription factors, it attaches to the beginning of a region of the DNA called the promoter, pries the DNA strands apart, and untwists a short portion of the double helix. RNA polymerase moves along the DNA, pairing up RNA nucleotides with their DNA complements, adding nucleotides to the end of the growing RNA molecule. Here's a close-up view of elongation. Only one DNA strand, the template strand, serves as a template for RNA synthesis. RNA polymerase moves along the DNA strand in the 3' to 5' direction, adding nucleotides to the 3' end of the RNA chain. Note that U in RNA pairs with A in DNA.
Once transcription is complete, RNA polymerase releases the completed RNA and detaches from the DNA.
So the DNA sequence where the RNA polymerase attaches is called the promoter region. Now, if we're talking bacteria, it's called the terminator at the end. Don't have that in eukaryotes. I don't think you're going to need to know that for the AP Bio test, though. And the stretch of DNA that is transcribed is called this transcription unit. That section basically where the gene is and a little bit of the information on either side to get things started, that is the transcription unit. All right. Let's get into the transcription details. So let's do a little more detail on the synthesis of an RNA transcript. Once again, you got 3 stages: initiation, getting it started; elongation, making it bigger; and termination, where do you stop making this thing? They start at the initiation point, at the start point. The transcription factors mediate the binding of RNA polymerase and the initiation of transcription. Now, the stuff I've got here later, I don't think you need to know for AP Bio, but I'm not positive, so that's why I've got it in gray. So all those transcription factors and RNA polymerase bound at the promoter, that whole thing altogether is called the transcription initiation complex. So it's a complex of things that are going to initiate or start transcription. And at the very beginning of this section, we have something called the TATA box, which you're going to find in eukaryotes. Let's show you on this image here. So here is that TATA box at the beginning of the promoter. This is the start point, and this is the template strand down here. This is the part we're actually going to use to make the gene, or this is the gene that's going to make the protein. The transcription factors will start binding on, finding that TATA box to tell them where the beginning is. That's going to then bring in the RNA polymerase, and this whole thing is called the transcription initiation complex. And we're going to start adding on here. On the DNA, we're going 3 to 5. RNA, though, because it's complementary, it goes opposite. We go 5' to 3'. Second step is elongation. So the RNA polymerase is going to move along the DNA, untwisting that double helix, 10 to 20 bases at a time. You're not going to need to memorize that. And we do about 40 nucleotides per second in eukaryotes. Now, a gene can be transcribed simultaneously by several RNA polymerases. So if this is my DNA here, I could be transcribing here, here, and here, all at the same time, transcribing 3 different parts of that RNA that we're going to make. And then they can be connected later. Okay. And like I said before, nucleotides are added to the 3' end of the growing RNA molecule. So here you can see we've untwisted it. 5' to 3'. Here we're adding on those new nucleotides, matching up A with T, U with A. U's kind of coming here with A again, making our RNA. But we need to know where to stop this thing, right? Otherwise, it's just going to keep on going, and it's going to just copy that whole strand of DNA until it gets to the end. We don't want that. We only want the gene for that one polypeptide that we're trying to make. And they're a bit different in bacteria and eukaryotes. In bacteria, it stops at the end of the terminator, a little terminator segment there, and then the mRNA is done. Don't need any further modification, like we said before. However, in eukaryotes, it's going to go until we hit something called the polyadenylation signal sequence. Basically, it's a stop making it here sequence, but that's the term for it, polyadenylation signal sequence. And that's going to tell the polymerase to stop adding these things on. Generally about 10 to 35 nucleotides past that sequence. Got an image of it. It's going to appear later, then also we'll point it out then. All right, folks, let's take a look at what happens to that RNA transcript after it disengages from the DNA, okay, or post-transcription modification. So, like we said, eukaryote, not prokaryotic, but eukaryotic cells modify their RNA after transcription. We've got a lot of enzymes in there in eukaryotic organisms that will modify that pre-mRNA or the RNA, and we call it RNA processing, before we send that out of the nucleus and to the ribosomes. During this process, both ends of the primary transcript, that initial thing we made off the DNA, are usually altered on the two ends, and often internal sections of the mRNA are cut out and the remaining parts spliced together. So let's talk about both of those points here a little bit more. So at each end of the pre-mRNA molecule, or the here, is modified in a particular way. The 5' end gets something called a 5' cap, or a GTP cap, or a guanine triphosphate cap. They're all the same thing, just different names depending on where you're reading about them. I've seen all three in AP stuff. The 3' end gets a poly A tail, so lots of A's, lots of adenines. And what we think those modifications do is help get the mRNA out of the nucleus and into the cytoplasm, kind of protect the ends so they don't get hydrolyzed by enzymes or start fraying and coming apart by the enzymes that are floating around in the cytoplasm. And they also help the ribosome attach to the 5' end, that 5' cap. It's a little like docking port for the ribosome, if you will, onto the mRNA. So we can see that here. Here's our initial transcript. Here's our 5' cap, or guanine triphosphate cap. Okay, we got our start codon here. We've got our polyadenylation signal we talked about earlier, and a little bit past that, that's where we ended our transcription, and we add on our poly A tail. Just lots of A's all in a row. Okay, like lots, 50 to 250. And then here we can see we got our start codon, our stop codon, and we've got different colors here. Some of these code for stuff and some of these don't. So let's talk about that. So most eukaryotic genes and their RNA transcripts have long stretches of them that are non-coding nucleotides, just gibberish. Okay, and then we have areas that are coding areas that actually code for making a polypeptide. Those non-coding areas are called interons, okay, because they're in between things. And the other regions are called exons. The coding regions, and I color coded here, right? Coding regions are exons because they eventually will get expressed and usually translated into an amino acid sequence. Not always, but usually. And then what we're doing here with RNA splicing, we're going to remove those interons, those non-coding things, and join the exons together so we get one long strand of mRNA that's ready to be transcribed in the ribosome. So here we can see, same one we had from before. Here's our start codon. Our stop codon was there. There's an exon, there's an exon, there's an exon. These are interons. Chop those out, put them together, and there is our finished mRNA ready to head out to the ribosome. This one I'm not sure on, guys, so I graded out. I don't think it's on the AP exam, but just wanted to include it just in case, because I heard about it other places, so I don't know. In some cases, RNA splicing is carried out by things called spliceosomes, kind of a fun name. They consist of a variety of proteins and several small ribonucleoproteins, SNPs, that recognize the splice sites. And they're like little scissors that go in there and cut them and put them together. And we take advantage of some of this architecture when we do various genetic engineering and things. Okay. So here we can see a spliceosome with the little SNPs in here doing their thing, cutting it here, and then joining them together to make our mRNA. And here's that cutout chunk. So why are these introns important? Why are they even there? Okay. Some introns seem like they might contain sequences that regulate gene expression, regulate when genes get turned on or off. Some genes can encode more than one kind of polypeptide, depending on which segments are treated as exons or interons during splicing. So the same gene could code for this polypeptide or that polypeptide in the same DNA, depending on which parts of it are spliced together. We call that alternative RNA splicing, which means there are more polypeptides or proteins that an organism can produce than there are genes in the organism, which is kind of fantastic. We already have so many genes, but we can still make even more proteins because of the way we can rearrange within those genes. Another interesting thing that's just fascinating with proteins and DNA and how we can get new combinations of things is that proteins often have a modular architecture. Modular, and as you can take this piece and this piece and put them together here, or you can take this piece with a different piece and put it together here and get different things out of it. We call those different discrete regions our domains, like Legos. Okay, well, you get a whole set of Legos and you got a bunch of subunits or domains. One of them might be a 1x6 Lego block. Another one might be a Lego wheel. Another one might be a Lego and satellite dish or something. Those would be the subunits. In many cases, different exons code for the different domains of our protein. So this exon codes for that 1x6 block. This exon over here codes for a Lego wheel. This exon over here codes for a Lego window. And so you got all these pieces you need to build your protein. Now, exon shuffling may result in the evolution of new proteins. So it's kind of like you've got your gene and you got these exons with these different domains that make different shaped chunks of protein, different functional units of what will eventually become a full protein. And you can follow the directions and make that protein do what it's supposed to do. But if you rearrange the exons a little bit, you can get it to make a different protein. It's got the same parts in there, but we're just rearranging the parts to make a protein that does something different. It's kind of like taking your Lego set and throwing out the directions and just taking all those subunits and putting them together in new and unique ways to take a space set and turn it into a castle or a house or a bantha or whatever it is you want to build. It's pretty sweet. Okay. And we think this has a big role potentially to play in evolution and making new proteins. We don't have to reinvent the wheel. We just have to reorganize the wheels in 1x6 blocks that we already have. So here we can see kind of an example of that. Here's exon 1, 2, and 3. Exon 1 codes for this domain, which would take this particular shape. Exon 2 codes for that domain. Exon 3 codes for that domain. And we could put them together like this, or we could put them together in a different way and get a polypeptide that works differently and does something else. And that will do it for 6.3 on transcription. In the next section, come back and join me where we actually build the protein in part 2C, 6.4 translation. See you then.